Welcome back, ladies and gentlemen. Mr. Hescox coming back to you. We have been in my life science class. We're working on genetics and soon heading into advanced genetics. We've talked about DNA, the structure of DNA, the bases, the sugars, the phosphates. We've talked about coding, A to T and C to G. We talked about codons and how those codons can code into an mRNA strand, which is transcription. Then we read that mRNA strand, which is translation, to build our proteins. So the question was, what is a codon? And a codon is three bases of DNA that code for eventually an amino acid. So today we're going to talk about mutations, and mutations are basically errors in DNA or changes. Changes in DNA cause variation. The differences in you or I are all because of little changes in our DNA. Humans are 99.9% exactly the same as far as genetics goes. It's that 0.01% that changes the differences in us. You have over 6 billion base pairs. 5% of those at any time code for proteins. Now, we can cause errors or changes in DNA because it just might, but there are things that can actually cause it, such as ultraviolet light, which you know causes you to get tan, but can also cause skin cancer. Different types of chemicals, gasoline and asbestos come to mind. Both of those, if you're exposed to them long enough, can cause cancer. And in radiation, different types of gamma radiation, alpha radiation, beta radiation are all problems at some times. You have your own set of DNA proofreading enzymes. And what they do is they are constantly searching your DNA looking for errors because errors can cause genetic disorders. The most problematic of all genetic disorders is cancer. So if you have an error in your DNA, it can cause that. Any type of change in the DNA is called a mutation. When it comes to mutation, there are 3 levels. So you have no effect. If there's no effect, the change in the DNA strand occurs in a non-coding region, meaning it doesn't code for a protein or some sort of trait. The effect could be minor. It may have like some deer may have a little bit longer hair than other deer. So that's a minor mutation. And then it could be a major mutation, such as we all know that deer can be albino. Well, a white deer does not blend in the woods, and they are at a greater risk of death because they stand out so much. Okay, so I have this article here, and we read this in class every year. This was a salamander that was mutated and found at Lake Racetown, which is what, 25, 30 minutes from here at the most. And some students at the local college there in Huntington found this, and you'll see here that it actually has a fifth leg fused to its third hind leg. And they talk about its spots and how each spot is different because we all have different genetics, but this genetic disorder here caused an extra leg to form. Other types of genetic disorders that we're going to talk about in this chapter are places where the genes cause something wrong. Alzheimer's, glaucoma was the disease of the eyes, diabetes. I've had several diabetic students over the years who can't produce insulin. Sickle cell anemia is a disease in which the hemoglobin forms incorrectly and it doesn't carry oxygen well, and it causes the red blood cells to form a sickle shape. And then, unfortunately, cancer, which so many of us are affected by cancer today. Whether you've had it or if you know somebody who's had it or lost somebody who's had it. This is sickle cell cancer. See how it's shaped like a C? There used to be an old farm tool that they would use for harvesting grain called a sickle. That looked like that. That's where it got its name. To get this, you have to have 2 bad copies of the gene because it's recessive. Normal red blood cells look like this. These don't roll and they get stuck in the joints. And I guess sickle cell actually is very painful. You can actually develop, you have one bad sickle cell gene, one good sickle cell gene, and you can show some of these traits, but it does help protect you from malaria. Like sickle cell does not get affected by the disease of malaria, but you have sickle cell disease. A pedigree is a special diagram used to track family relationships for 2 or more generations. You often hear about pedigrees with dogs or racehorses because they want to track the championship breed. We can use pedigrees as humans to track genetic disorders. This pedigree you're seeing right here is of the royal family of England. This is Queen Victoria of England, and she was a carrier of a sex-linked disorder known as hemophilia. Hemophilia causes you to bleed uncontrollably. Basically, the stuff like if I get cut, eventually I'll get a scab and it clots. If you have hemophilia, it will not clot right. Now, hemophilia is sex-linked, so it's carried on the X chromosome. The X chromosome is the only chromosome that in men is not paired, because in women you are XX. So let's say that you have a gene for hemophilia. The normal gene takes over. If you're a man and you get one gene for hemophilia, the Y does not have the correct enzymes or proteins or, genes to override that. So here's Queen Victoria and Prince Albert. Now they had 1, 2, 3, 4, 5, 6, 7, 8, 9 kids. 3 of the girls were carriers. That means they had 1 bad gene for hemophilia, 1 good gene. Poor Prince Leopold actually had hemophilia. So that's her first round of children. Now in her grandchildren, she had 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 14, 15, 16. She had 16 grandchildren. The circles are girls, so this one's a carrier, this one's a carrier. All of the red squares are boys who actually had hemophilia. Every single generation becomes more and more prevalent. And if you want to get down to the modern royal family, they're over here. Here's Prince Philip, Queen Elizabeth, who's still waiting. She is still queen right now. The next to be in line is Prince Charles. He was right here. There's Princess Diana. She died in a car wreck. So we're going to finish up talking about mutations. I hope you learned some stuff. We'll talk more about mutations and genes in the next section. Until then, we still have to kind of cover here cancer. Cancer is a really tragic genetic disorder where the switch to turn certain genes off becomes broken. So every cell has the genes that tell it when to start to divide. There are also genes that tell it to stop dividing. If cells become injured, we divide, we make new cells to replace the ones that are injured. Another gene tells those to stop. In some cases, the switch that tells the gene to stop is broken, and that allows those cells to divide and divide and divide and divide until you get these massive cells that can actually kill the cells around them because they're dividing and growing so fast, they take all of the nutrients away from everything around it. And then when cancer gets really crazy is when it spreads. The spreading of cancer is because these cancer cells can break off, and they'll travel through the blood system or through the lymph nodes to other parts of the body, where if they get lodged in another part of the body, that cancer cell will start to grow again and divide and divide and divide. So if you hear of a person who has had cancer that spread, that means that they had a piece of the cancer broke off, traveled through their body, and started to grow in another area. And that's when cancer is really dangerous. So carcinogens are chemicals that cause cancer. There's so many different types of chemical carcinogens out there. Formaldehyde, which is, if you ever really want to know how many carcinogens there are, just read the list of carcinogens in a cigarette or in one of those vapes. There's 90 to 100 different carcinogens in a cigarette or a vape pen. Ultraviolet, X-rays, all cancerous as well. So cancer by far is the major problem with human mutations, but there are so many genetic disorders that have been caused by mutations over the years, and if you're more interested, I'd be happy to talk to you more. But until then, be good, be safe. I'm Mr. Hescox. See you later.